So we are Corentin and Charlotte from Decision and Bayesian Computation Laboratory at the Institut Pasteur. And we are going to present you our project of visualization and analysis of large microscopy images in virtual reality. So first of all, the major challenge with 3D microscopy images is the lack of standardization. Indeed, these kind of images depend on the biology of your element of interest, but also on the hardware, the microscope you use to generate your image. Finally, it also depends on the protocols for the sample preparation. The other challenge is the poor suitability of the existing visualization software, which are limited to a 2D screen and require some pretreatment. Additionally, this software has a really poor data interactivity. In order to answer to this challenge, we developed in the laboratory an interactive and high-quality visualization tool called DIVA using the virtual reality environment. We combine this software with a one-shot learning and feature extraction in order to do some fast 3D segmentation. Now I will present you the DIVA software. So DIVA stands for Data Integration and Visualization in Augmented and Virtual Environments. DIVA is based on a dual interface. So the first interface is a desktop interface, as you can see on the left, where your image stack is directly loaded at the 3D volume. You can interact with this volume simply with your mouse, and you can also make some real-time modifications, such as the color and transparency of your voxel. Once this visualization parameterization is done, you can simply see your image stack as a physical object in front of you thanks to the VR headset. In this virtual environment, you can do some real-time manipulation with the VR controllers. These controllers can also be used as a clipping and counting tool. All the measurements can be extracted as a CSV files.
I will now drive you to different steps of the voxel learning pipeline for 3D segmentation. Once the original image has been loaded in DIVA, here you can see our working example collected in a collaborating lab from the Institut Pasteur. You can see mouse olfactory bulb interneurons collected on a confocal microscope. The first step in virtual reality is the data tagging. You use the virtual reality controller to tag voxels, only a few of them, with two different colors, blue for voxels belonging to a neuron and pink for voxels not belonging to a neuron. From those voxels, DIVA will then extract 31 features calculated using different spatial filters, as you can see, for instance, Gaussian filter or mean filter. Those features for the tagged voxels will be used to train a classifier that will output a classification probability indicating whether the voxel belongs or not to a neuron. Consecutive to this training phase, we will now show the inference phase. DIVA extracts features for every voxel from the original image and run them through the classifier to perform inference and output probability for every voxel to belong or not to a neuron. You can see the results on the top right corner of this slide. To optimize the computational resources, as the virtual reality is very demanding, especially in GPU, we implemented a solution with cloud computing to outsource the training and the inference parts. To enhance the robustness of our final classifier, we added weak classifiers whose output probabilities were added to the list of features. We can as well iterate the whole process to refine the segmentation in what we could call a correction tagging step. Here you can see the two different applications of our voxel learning pipeline. On the top, it's the full images from which our working example was drawn. You can see that the segmentation can easily propagate to the full volume. On the bottom, it's a different examples. It's mouse microglia image here with a 2-photon microscope. You can see that even though it's not the same structure, the segmentation is perfectly conducted as well. From these results, we can extract some relevant information. First, it can be used as a direct image preprocessing after acquisition. We can use it as well to count or measure structures of interest, or to extract some morphological information.
To conclude, DIVA offers an interactive environment in VO in order to better visualize some 3D microscopy images. In addition, the one-shot learning allows a quick and flexible segmentation without database. Finally, cloud computing enables a fine allocation of computational resources. We can find DIVA in the website diva.pasteur.fr.